Hello everyone and welcome back to Chapter 18, Lecture 3. We are moving on to the second part where we will finish up the thyroid gland, and we're going to talk about some of the higher detailed stuff like something like thyroid hormone production, as we're going to see. And then we'll see that there is an embedded gland. There's actually 4 of them called the parathyroid glands, and then we're going to relate all of this to calcium regulation here, which is a big concept. Just like blood glucose regulation is huge, you should be thinking pancreas when I mention that. Well, with calcium regulation, we're going to rely on the thyroid gland and the parathyroid gland. If you remember, during part 1, I sort of mentioned those C cells, as you can see in the histology slide here. These are those little C cells in the middle there. They secrete something other than thyroid hormone, which thyroid hormone should be stored in the colloid, as we can see here. You can see more C cells on there. And then we have, of course, Pearson's wonderful figure here. As I continue to encourage you, make sure you're using those figures. If you can explain the figures, you likely know the content. If the figures don't make sense, you're probably missing some key points, and that is a really simple truth, a really simple strategy that I would encourage you to make use of. Alright, so here are the objectives. As you can see in the first part, we really just got into the anatomy of the thyroid and talked about the functions of T3 and T4. Well, we're now going to expand on T3 and T4 production, and then we're going to talk about some imbalances. So we're starting to get into imbalances. We've already seen that with diabetes, of course. We've talked about growth hormone, so we're continuing that sort of factor. And then, as you can see, as we move on to part 3, we'll continue that and talking about kind of the dysfunctions as well with the adrenal gland. But anyway, let's move on and actually get into the lecture here. So we're moving on to the formation of thyroid hormone, and this is one of the more detailed processes. So the big picture concept is this is how many different hormones are formed. So we're looking at kind of an extra step because most glands aren't going to have a colloid like this. So we're going to see some of that sort of formation that's unique. But ultimately, the concept is fairly similar on how certain hormones would end up forming, which is basically get your ingredients in, combine them, do a bunch of chemical reactions, break them down into individual units, and then release them. So let's start by explaining this. So that's the big picture. That's why you're starting to learn this. And we're looking at basically a process of protein formation is really the way to think of this, and the steps in essentially forming the peptide hormone, or in this case, since it's thyroid hormone, we know that this is a tyrosine derivative. But the concept of forming the complete formation of a protein or a molecule is being illustrated here. So that's the big picture. All right, so let's get into thyroid hormone formation. And so step number 1 is those iodine ions are going to be actively transported into the follicle cells. If you remember, one of the key points is that ion pump is TSH sensitive, meaning thyroid stimulating hormone sensitive pump. So step 1 makes sense to say, hey, when the TSH binds, it's going to cause iodine to be absorbed. So again, get your ingredients that you need to form the molecule into the cell. Step 2, you need to know the follicle cells, and you can see this is a follicle cell. As I pointed out, you should know that these are follicle cells because there's also C cells. You should know that they are going to produce or synthesize thyroglobulin, right? That's that TGB. And so you can see thyroglobulin, or TGB, is getting produced there. And all that was, if you remember, is a tyrosine globular protein, and it's the thyroid hormone. That's what this TH stands for, right? TH, thyroid hormone, TH abbreviated, so this stands for thyroid hormone. It's a thyroid hormone precursor. It's the starting material. That's all you need to know. Basically, it's a blob of tyrosines is the way to kind of think of it. It's a blob of tyrosines, but remember, we're talking a tyrosine derivative. We don't want a blob. We want individual units. All right, so steps 1 and 2 should be pretty clear. Just get your ingredients that you need. So the follicle cell produces TGB, and then we do get our iodine in. Now, if you were to argue and reverse these steps, I don't think that would necessarily be incorrect. You certainly can argue that TGB gets produced and iodine ions come in later. But we're going to follow this specific order. And the reason I'm saying it could be reversed is because, truly speaking, the cells are producing TGB and cells are bringing them in. So what happens first? We're going to start with iodine being the first part, and thinking TSH activated the iodine ion pump, and then that stimulated the cell to start producing TGB. Step 3 is we're going to have TGB get iodinated. See right there, tyrosines of TGB get iodinated at the apical surface. So one of the big things that I stress on this is you need to know where this iodination process occurs, and that occurs at the apical surface. So that's one of the individual questions that I ultimately can single out when I test. And so when I test my students, I often will single out, hey, where do the tyrosines, or I should say, where do the tyrosines of TGB, where do they get iodinated? And they get iodinated at the apical surface. So I expect you to know it's within the follicle cell at the apical surface. Now, once that process happens, we now just ship the iodinated TGB. It gets exocytosed, that means it gets removed, but it's going into the follicle lumen space or into the colloid. So if you want to put it's within the colloid, that works as well. In fact, you'll see on my bullet point it says within the colloid this occurs. So notice, form your starting materials, combine your starting materials, now ship them into a new location. And the reason for this shipment into the new location is there's going to be various enzymes in the follicle that are not found within the follicle cell. So within the follicle, which is the open space or the follicular cavity, in there we have the colloid, and that colloid is going to contain enzymes. Those enzymes are going to allow chemical reactions to occur to form globular T3 and T4. And what that simply means is we still have a blob of tyrosines, but those tyrosines are now no longer the precursor, the thyroid globulin. They are now actually functional T3 and T4 at this point. Now, most of my, just to be clear, most of my slides are color coordinated, so purple would match up with purple, but in this case, it's not. The colors are just changing to illustrate what each step really the key point is. So this purple does not line up with step 1, it's just its own singled out thing, and so the remaining colors, if they repeat, they're not overlapping with one another like most slides tend to do. All right, so step 6, that globular T3 and T4, that are formed within that globular TGB, essentially, that gets endocytosed. And so at this point, it's really, notice, thyroid globulin containing T3, T4. It's basically the functional unit getting pulled in. Now that we have the functional unit, we have functional T3, T4, we now have to break them down. So again, different enzymes that exist in the follicle cavity will now break them down, specifically the lysosome. If you remember, that's an organelle that digests things. That'll break that globular protein down into individual T3, T4 hormones, which again are tyrosine derivative hormones here, but still lipid soluble. And then step 8, if you remember, anything that's lipid soluble has to be a bound hormone, right? We talked about that concept that things that are lipid soluble, they need to bind to things. And so you should know the final step is T3 and T4, they're going to bind to thyroxine binding globulin. So that's the specific protein I expect you to know. Otherwise, if I was doing this as a stepwise fashion, I could just say T3 and T4 bind to a transport protein. So either way, it's fine. You should know thyroxine binding globulin is the specific one, but you can also notice this is thyroxine binding globulin at this point, so TBG. But notice there's a few others. There's albumin. You don't really need to get caught up on them. Like I said, usually I just kind of stay, if I'm going to summarize this, I'll say something specific about binding to a transport protein. All right. Notice I spent a lot of time on this. This is one of your more detailed things. So for my students, you can expect at least one question just knowing these steps. And if you notice, I had 8 steps where the book only had 7. I always stress this with students. I know everybody likes things being crystal clear. Hey, this is the number of steps and there's no changing it. The reality is, it's not about the numbers, it's about the description of the process. So if you wanted to break things up into individual steps, you can actually have 10, 11, 12 steps if you wanted to break these individually. Or if you wanted to combine steps, you can shrink them down to down to like 4 steps. It doesn't matter. Any process that we describe, you can do this. It's not about the number. It's about knowing the process and the events that are occurring. So I have 8 steps because I do a couple things. Like notice my step 3, I talk about them getting iodinated at the surface. Notice their step 3 is actually my step 4 where this chemical reaction starts. So I actually have that step singled out. Now, individually from this, so when you're studying this, yes, it's great to know the steps and be able to put them in order, especially on a multiple choice question exam. But for me, I also, like I said, I can also ask, where do tyrosines get iodinated? Well, you need to know the apical surface inside the follicle cell. You also need to know, where does T3 and T4 form in their globular state. Well, that event, the formation of T3 and T4, that occurs in the follicular cavity or the colloid. Either answer is good. Usually I put colloid. And then you need to know where actual individual T3 and T4 form. Those are going to form specifically within the follicular cell itself. Okay, those are how I single out some concepts. So that's what I would encourage you to focus on when you're breaking this entire process down. And after describing a complex process like this, I usually do a simplified version of this. And you're going to see that it ultimately ends up being the same steps. This is the detailed side, and the reason I say this is detailed, like notice iodine ions actively transported. That means we were going against the gradient, that there's more iodine in here if we're using a pump or literally forcing iodine in there. Follicle cell synthesized TGB. Do you know what thyroid globulin is? Right? That's the detailed side. So the simplified version, if you notice, is get iodine, form TGB, combine iodine and TGB. That's your first 3 steps. Notice no details to it whatsoever, but that's truly your 3 simplified steps. Then now you have your TGB and iodine combined. What do? Well, if we think about the next steps, we transport the TGB into the colloid, and then those enzymes convert TGB into the globular T3 and T4 within the colloid. Okay, so those are kind of the quick summary there. Then the next step is that we bring it back in and we finish the processing. So globular T3 and T4 are endocytosed. That means brought back in. Lysosomes break them into individual units. Now that we have individual units, what do? We ship them out into the blood, and then they bind to a thyroxine binding globulin or a transport protein. So literally, if this is why I say you can almost think of this as 4 steps. What happens initially in here, what happens in here, what happens in the second time, and then once it reaches the blood. And that's kind of how I would view it. It's 4 steps that you kind of want to summarize in sort of this extent. I would encourage students to start here and really work on the simplified version, and then see if you can go back and make sense of the bit of details to it. This is going to take you some time to break down and break through. Like I said, on an exam that I usually have close to 100 questions, figure that there's going to be about 3 to 4 questions coming from this. So if this is causing you a lot of stress and confusion, what I would encourage you to do is just read over it every time you study. Just read through it once. Don't spend too much time on it. And then once, at some point, usually around the 4th or 5th time you read it, some things will start to make sense and you'll start to understand the figure. If that and when that happens, that's when I would start actually actively studying and spending longer time on that. Until that happens, where you can read through this and look at the figure and say, oh, I'm starting to make sense of it, I would just constantly read through it and then move on. And I know that sounds weird to say, hey, I'm not going to spend too much time and really critically think about this, but tough concepts, if you get in the habit of just trusting that you read it, and then some things will start clicking, that is what you should do, and that'll make it a lot easier to study it later on. Right now, this is probably really overwhelming. It was a ton of details that went into this, but like I said, focus on breaking it down more on the simple side, and then once you kind of make sense of the figure or starting to see that the figure is making more sense, then actually take your time with it. But until then, like I said, it's 4 questions, so it's enough that you should want to study this, but at the same time, you have a lot of other areas to focus on. So do a fast read through every time you start to study. Just read through the steps, and once it clicks, start studying longer. All right, but let's move on to the next part. So with the thyroid hormone, so now we really work towards applying our knowledge and our understanding of content. And so that comes from the imbalances. And if you understand the imbalances, that means you understand the hormone, and vice versa. If you understand the hormone, you should be able to think, what could these imbalances lead to? So these are really great. I would make sure you study these. Anytime I talk about things in my lectures where there's disruption to normal homeostasis, it's definitely a concept that's highly likely to be tested on. All right, so thyroid hormone imbalances to be aware of. We can have something called hyperthyroidism. And when you see the word hyper, just like when we think of kids that are hyper, we think they have a lot of energy. So hyperthyroidism means we have high levels of thyroid secretions. And so if you just think back, what were the main points of the thyroid hormone when we talked about it? Well, it was elevating oxygen supplies and energy, right? We talked about important for growth and development, and we talked about important for maintaining body heat. Those were like the three major points that if you didn't get it down with thyroid hormone, I would make sure you write those down. Important for energy processing / metabolism, important for development, especially in kids, and then important for heat maintenance. Now notice hyper, meaning we have high levels of thyroid. That means we would think the person's super active. They're probably going to be hot all the time. Okay, and those are the things we should expect to see. So notice increase in metabolic rate, high body temp, weight loss, because that's increased energy, right? Increased appetite, that's going to relate to using energy, lots of sweating, rapid heart rate, high blood pressure. All of these things should make sense, all right? So now we can go to weak muscles. We can go to hyperactivity. That should make sense. The weak muscles is a little bit tough to understand, but again, you have high metabolism, so your muscles are breaking down and utilizing energy. This is not typically a major point that I would definitely stress, but if I stress things like metabolism, body heat, as I've stressed, these you would expect to minimally see. Increased body heat, increased metabolic rate. Usually I mention weight loss and those sort of things. So being able to recognize that this represents hyperthyroidism. All right, and with practice, there's a few activities we do in class. With those activities in class, plus having these notes, you'll see hyperthyroidism is very, very distinct in comparison to a lot of the other imbalances. All right, it's not very similar to diabetes at all, right? So far we haven't seen too many overlapping imbalances yet. It's not anything related to growth hormone either. The flip side, if we have hyper, we can have hypo, and hypo means low levels. All right, so low levels of thyroid secretion. So again, if you know what the thyroid hormone does, if you have low levels, just kind of flip all of these symptoms. So if you want to think about what these should be and maybe come up with some answers, pause the video for a quick second. But I'm going to go ahead and play it along here. And so hypothyroidism, the symptoms are decrease in metabolic rate. Notice low body temperature. There's likely to be weight gain, a reduction in appetite, low sweat and oil secretions, which can result in dry skin, reduced heart rate and blood pressure. Now, there's a enlarged heart. This is, again, one of those weak muscles sort of thing that I don't tend to focus on, but this is related to hypothyroidism. There's sluggish movement with weak muscles, all right, apathetic, always fatigued, and other symptoms. If you know anyone who deals with a thyroid issue, it's really, it needs to be, this hormone needs to be maintained in a really tight range, and if not, it causes pretty dramatic swings in an individual's weights and individual's mood and energy levels. It's just such a critical hormone to have in balance. So you can see from these two things. And so if somebody has an issue, it's usually swinging from both hypo to hyper and vice versa and trying to get a good balance. I worked in the pharmacy for a little bit, and what was interesting about these medicines, most medicines are in either grams or milligrams, and a milligram is 1,000th of a gram. The thyroid hormones were in micro, which is 1,000th of a milligram. So we're talking, what would that be? A millionth of a gram. So really, really low amount. And the fluctuations were like 50 micrograms to like 66. I mean, they were really short distances from one to the next. And so that's how tightly regulated this hormone needs to be. All right, just to give you some interesting facts there. Now, with the thyroid gland, so this wraps up thyroid hormone, but we do have one last thing, which were those C cells. And so here, I think this figure, which is borrowed from a different book, this is not Pearson's, this I believe is McGraw-Hill's, I think I borrowed it from there, but you can see here we have a follicle cavity, or a thyroid follicle, and I think this does a nice job showing you the spherical design when you're looking at a histology image like this. And don't forget, listen to the lab lectures if you need to know the specifics for what I expect you to know on the lab related to histology, and histology was covered in the previous part. So lecture number 3, part 1, did cover some of the lab concepts, but we're going to see a few of those here as well. All right, but the C cells, these are going to produce and secrete calcitonin, and calcitonin was covered last semester, or the first semester of A&P, when we talked about the skeletal system. And hopefully you remember what calcitonin did, but if not, the things that I stress is calcium in. All right, so calcium in. And so basically what calcitonin is going to do is when our blood calcium levels are high, calcitonin gets secreted. And what it does is it really regulates the blood calcium levels and within the body fluids. And what it does is it inhibits the osteoclast, which isn't the big thing. The main one is this. I would circle this over and over because I like the stimulation side more so than the inhibition side. Certainly it inhibits the osteoclast because those are the things that break bone down and would release calcium. But notice it's going to stimulate the osteoblast. To me, that's the important part to recognize because this is going to increase the uptake from the bloodstream, which means ultimately calcitonin is going to cause a decrease in blood calcium levels. All right, so it regulates calcium levels in the blood and body fluid by stimulating the osteoblast. Osteoblasts are the cells that are going to use calcium. So they uptake calcium from the blood, and then we increase excretion of calcium at the kidneys. So we're literally urinating out any excessive calcium. So the net effect of calcium in the blood, you really need to get down, and this is something I test heavily on, is it's going to decrease the amount of calcium in the blood. That's the main point. Decrease in available calcium in the bloodstream, so lower blood calcium levels. And notice I'm using various terms to say reduce, lower, etc. You need to be really good with perspective and using appropriate terminology here. So your C cells are these clusters over here. Like I said on histology images, I can circle various cells and ask you what they are. These would be all the follicle cells. So if I circled this whole thing and I would say, what are the names of the cells that form this structure? You would say follicle cells. Otherwise, I would circle in regions like down here, what are these unique cells or what are these cells here? Notice there's no colloid. If there's not a colloid formed, you know that it's going to be the C cells. All right. So main points of calcitonin is calcium into bone. So that means the osteoblasts are going to use the calcium that it takes from the bone. And then you have to remember, excessive concentrations of things can get removed at the kidneys, and that's what we see here, excretion at the kidneys. All right, so that's going to wrap up the thyroid gland as a whole. But before we finish this, on the posterior side of the thyroid gland, you should know we end up having what's called the parathyroid gland. So these are the parathyroid, and they're embedded in the back. And so notice it's a large purple-like structure, and so histologically, you should be able to see the follicles of the thyroid gland, and then you should just see this dense purple region. The dense purple region here is the parathyroid gland, and the only expectation I would have for you to know is it's going to increase blood calcium levels. So if I ask what's the function of the hormone it secretes, because notice it's the parathyroid gland, and that parathyroid gland secretes parathyroid hormone. It's not likely that I'm going to ask you what's the hormone secreted by this, but likely going to ask what does that hormone do. So parathyroid hormone is secreted when blood calcium levels are low. So that's when it's secreted, where when we talked about the C cells producing calcitonin, that was when blood calcium levels were too high. Parathyroid is secreted when blood calcium levels are too low, and therefore how would we get more calcium back into the blood? Well, it's the same two targets. Instead of targeting the osteoblasts, we're going to target the osteoclasts while inhibiting the osteoblasts. So we stimulate osteoclast activity. That causes the breakdown of calcium off of bone, and you should note that overall parathyroid is considered calcitonin's antagonist. Okay, and we talked about synergist and antagonist with muscles. Same thing with hormones. If you have hormones that really do the exact opposites of one another, you can call them an antagonist, or if they help each other out, you can call them synergists. But in this case, this is an antagonist because it's the exact opposite. Stimulating osteoclasts where calcitonin would have stimulated the osteoblast, enhancing calcium reabsorption at the kidney, and what this is saying here, again, this is parathyroid hormone, it's increasing the amount of calcium at the kidney. So instead of letting your kidneys release calcium into the urine, the kidneys, as urine formation happens, the kidneys are going to pull out that calcium and make sure it does not make it to urine. All right, but the third and final impact here is it's going to stimulate the formation and secretion of calcitriol, which you should know calcitriol is produced by the kidneys. It is a steroid derivative hormone, which is pretty unique. Now, what's interesting is calcitriol complements PTH, and what I mean by that is it is going to target the osteoclasts. All right, so it's going to stimulate the osteoclasts as well. However, because it's released at the kidney, it's not going to cause calcium reabsorption at the kidney. Instead, it's going to cause calcium absorption at via digestion, so at the intestines. You can say via digestion, and off to the side, I would encourage you to write at the intestines. So calcitriol does, number one, stimulates the osteoclast and inhibits the osteoblast, but calcitriol, instead of targeting the kidneys, it'll target the intestines instead. Make sure you know the difference. I fully expect my students to know that subtle difference between them. And then, just like how this parathyroid hormone was calcitonin's antagonist, calcitriol is actually parathyroid's synergist. All right, and so here's the calcium regulation right here, being able to break this down. And so if you notice right there, here they say PTH, increased calcitriol production causes calcium absorption at the digestive tract. So that's because of calcitriol. Again, this increased reabsorption of calcium at the kidneys is not a factor of calcitriol. It is strictly PTH. Make sure you know these really well. You know, big picture wise, parathyroid, what if I ask generically, what's the role of parathyroid hormone? It's to regulate calcium ions. What's the role of the C cells? Regulate calcium ion concentrations. Now the how explains the detailed side. So calcitonin, when calcium levels are high, calcitonin is secreted to decrease blood calcium levels by causing it's going to decrease blood calcium levels by causing the osteoblast to absorb the calcium and cause the kidneys to push out calcium into the urine. PTH does the exact opposite. PTH stimulates the osteoclast to break down calcium from bone and cause the kidneys to reabsorb calcium, which increases blood calcium. So PTH is secreted when calcium is low in the blood, and then that PTH will stimulate the release of calcitriol from the kidneys, which, similar to PTH, which is stimulating those osteoclasts, but instead of targeting the kidneys themselves, it will actually cause absorption at the intestines or via digestion. So know these three really well. I cannot stress that enough. It's a good thing for me to test on synergists. It's a good thing for me to test on antagonists. And then those subtle details are super, super important. So make sure you get these down. Again, calcitriol is one of your steroid derivatives. PTH and calcitonin are still part of your peptide derivatives. All right, so this lecture went a little bit long. We're right on the cusp of 27 minutes here. Obviously, a highly detailed lecture. That's why it took a little bit longer to get through. So part 2, certainly one you're going to need to spend a bit of time on. We'll be moving on to the next part, which is going to take us through the adrenal cortex. And there's roughly 3 major regions, so that one's going to take a bit to get through as well. But we're getting through the vast majority of the lab content so far. Again, because in my classes, the lab exams come before the lecture exams often, especially if it's hybrid. That's usually how I do it. So if that's going to be the case, if you know the lab exam is coming up before lecture exam, make sure you've started listening to the lab lectures. Otherwise, if I've said we're going to do the actual lecture exam first and then the lab exam, well, then continue to focus on the lab, the lecture side. But in most cases, I know I tend to do the lab stuff first. All right. Take care, everybody. Look out for the third and final part that'll finish out lecture 3. I think it's a little bit simpler because we don't have the formation of a hormone. And then we'll be wrapping up with the fourth and final lecture of the chapter 18 here, which will go into that lipid-soluble versus water-soluble and how those hormones function. So we'll be wrapping up with a few tough concepts in the near future. Take care, everyone. I'll see you at the next part. Thank you for listening and following along with us.